So, hello guys, and today we'll be continuing with the transcription of in prokaryotes. And so, in the last class, we studied about the in the last class we studied about the transcription and basically transcription in prokaryotes. Okay, and the various steps involving initiation, elongation, and termination. And we also studied about the promoter, the RNA polymerase, and the different antibiotics that are used to basically stop either of these steps so that we can use it in treatment. Okay, so now we'll be moving on with one more concept. Okay, so we'll be studying about how we can, starting with this question, we'll be moving on to the regulation of gene expression. So this question is, through which technique RNA polymerase binding to DNA can be detected? So basically this question is related to DNA protein interactions. So how we can detect any of these DNA binding proteins that are binding to basically different regions of DNA. So here are some of the techniques mentioned, like DNase 1 footprinting, enzyme mobility shift assay, chromatin co-immunoprecipitation, and all of the above. So basically all of the above is the answer. So we can detect like the DNA protein interactions or a protein that is being bound to a particular DNA molecule through either of these techniques. There are more also, but we'll be more focusing on these three techniques. Okay, so starting with the first technique, that is enzyme linked mobility shift assay. So what is this? So first, let us consider that there is a DNA molecule. So for example, this is the DNA molecule, and this is the protein molecule that is being, we are testing whether this protein is basically bounding to this particular DNA molecule. Okay, so now what we'll do is we will have three samples. Okay, so uh, like we will make the three cultures. So this is an in vitro assay to detect any of these. So we will purify the DNA, the pure colonies of the DNA that we need to test, through which the protein is binding, and the pure solution of the protein, of which we have to check the interaction. So we'll make three samples or three solutions. Okay, so first one would contain only DNA, in which protein is not there. Okay, in the second sample, what we have, we will add DNA plus the protein that we need to test whether it is interacting with the DNA or not. And in the third well, if we have to interact if we have to check whether more proteins are being bound to this particular complex, then what we'll have, DNA plus the protein, in consideration the main protein that is binding to the DNA, and after that the protein A or any other protein that you have to check whether this particular protein is being bound to the this particular complex. Okay, now what we'll do, we incubate it for some time. For example, we incubate at 25 degrees Celsius or 37 degrees Celsius based upon the interaction. Generally 37 would be preferred because it's the normal physiological temperature. Okay, and then we'll incubate for some time, and then what we'll do, we'll run on a agarose or polyacrylamide gel. Basically agarose would be preferred. to check how this particular thing will be seen. Okay, so now what we'll see is like in either of these wells. In the first well, we will see that only the DNA molecule is present. So we will observe the band at a particular position. And in another well, we will have ladder also. Okay. So this ladder would be nucleic acid or DNA ladder. That will be most preferable because we are checking the mobility of DNA molecules here. Okay, in the second well, we will have the complex of DNA and protein. So the unbound DNA that will have will be formed in the below. So the band will be observed in below. And if we observe a band upper to this particular thing, in this particular band, then that means that DNA has bound to the protein. Okay, so why am I saying is, so whenever if this particular DNA molecule is being bound by a protein, that means its molecular weight has increased. Okay, so and as we know on a gel, if we run a particular column from negative to positive, so what happens? The DNA molecule being negatively charged, and depending upon the size of this particular DNA, the smaller the DNA, the farther it will move, and the larger the DNA, the slow and limited it will move. Okay, so if this would have the smallest molecular weight, so it will move more towards the positive end, and this will have the large molecular weight, so it will stay on towards the negative end. Okay, so this means it moves slower, and this moves faster and farthest. Okay, so this same principle is applied in the this particular essay also. So here the band is formed above this particular thing. So here the molecular weight is high. That means the DNA has interacted with that particular protein that we have added in the second one. And then if we have to check the presence of more proteins, so what we like if there is a band above also, that means that it is binding to more proteins, leading to increase in molecular weight. Okay, so if a band would be observed in like how can we detect that more than one protein molecule is being like interacting with the same DNA molecule? So if like it is interacting more than uh, twice or thrice in homodimer or homotrimer, so what we can see is another band could be observed before this band. So that would like give us an idea that okay, this particular protein is being like binding to the this particular DNA molecule in dimer or trimer. Okay, so this is the basic principle of enzyme linked mobility assay. Okay, so, and yes, how will you detect it? We'll detect it because the ends of the DNA is being labeled. Okay, so using autoradiography, we can detect the DNA bands that are being bound to the protein. Okay, so this was the paper which used this particular technique, and this was the probe that we used, and this was the increasing concentration of the repressor. So this particular paper refers to lac repressor, which we talk about in like in a couple of slides. And this lac repressor is being bound to the operator molecule. So as we increase the concentration of the repressor, the above band has increased as compared to the lower band. So here the repressor is being bound to the probe molecule, and this one is the free probe. Okay, so so it is like the repressor bound probe will move slower as compared to the free probe. So this is the basic principle of EMSA. Okay, now moving on to the next technique, that is DNase 1 footprinting. So what happens in this case? if a particular DNA, so like we considered a DNA, okay, and if a particular like a protein molecule is being bound with this particular DNA molecule, so if we treat this particular complex with any of the nucleases, okay, here we are using DNase 1. If we are treating it this particular complex with DNase 1, then what will happen? The protein that is being bound to this particular region will be masked, and here no DNase molecule can act, because protein is being bound to the DNA molecule and it's protecting the DNA molecule from the action of the DNase 1 and the rest of the region that is present here. So from starting from here to here, so all the regions will be cleaved off by the DNase 1 protein. Okay, so here as you can see, this is the DNA binding protein region, and here there is no DNA binding protein. And these are the basically areas where DNase 1 has cleaved that particular DNA molecule. So first, in this case also, we label the DNA that we are considering, that the probe that we are using must be labeled. That then only we can identify on a gel that how it is cleaving the DNA. Okay, so then there is no protein, and in this case there is protein that we have added. This is the protein binding sequence where the protein will be bound. So in one case, after binding the protein, when we add the DNase 1, it will cleave on the regions that are not bound by the protein. So in this case, no protein is there, so it will cleave in different fragments. And in this case, it cannot cleave in in this particular area because the protein is already bound to the DNA molecule. Okay, so after this, after the reaction is complete, we'll run it on a gel with the ladder, and then what we found is that in case of the uh, footprinting tail, so when the DNA protein is not present in the DNA, then what will happen? This particular like type of bands will be formed, and then if we add the particular protein in that particular DNA solution, then this band pattern is observed. So any band that is missing from the negative ones will show us that okay, this region is there that is being bound by the protein. Okay, so this is called as the footprint of that particular protein. And now what we can do is we can just sequence that DNA sequence using various sequencing techniques, so that we'll get to know the correct sequence of the protein binding site. Okay, so this is how DNase 1 footprinting works, and this is the gel that we saw in the like in last slide also. So this is how it looks, and most probably the third and the fourth well, because third well also have some band that are lighter than the second one. So this would have either like less quantity of protein, or the protein is being degraded using various methods, and the fourth one has the intact protein, so there are no bands formed in this region. So this shows that okay, the protein is being bound in these particular sequences. This is the technique of DNase 1 footprinting. Now similar technique is there, that is DMS footprinting, so dimethyl sulfonate footprinting. So what we do is it like it is also based on a similar method, like the protein will mask the DNA. So what we use is, here we use dimethyl sulfonate, okay, or DMS. So what it does is it will methylate the basically So here, what happens? The DMS first methylates the purine bases. Okay, so it methylates purine bases. So what will happen if the protein is bound to that particular sequence? Then this purines will not be methylated in this particular sequence, and the rest of the purines will be methylated. And what will happen? This has also the tendency to break all the methylated purines. Okay, so if we have like any chemical compound that can break the bond of the DNA where the purine is methylated, then after that we'll add that particular compound, and then what we'll have is we have different fragments of different sizes who have which ends are labeled, and then we'll run on this particular gel, and we'll found out that okay, so if there is no protein, then this particular band pattern is observed. If there is like one part of the protein added, then this particular band pattern is observed, and if the five parts of the protein is added, that means this particular band pattern is observed, and this is different from the one that is being in which no protein is added. So that will give us a particular footprint. Okay, in this region the DNA is being bound to that particular like protein is bound to that particular DNA molecule. So this was the another footprinting method through which we can detect the various like protein molecule is being added to that particular DNA molecule. Okay, now moving on to the chromatin coimmunoprecipitation or chip. So what we see is here we have a protein that is being bound to the particular DNA molecule. Okay, so now we have this particular protein. So we first what we do is we make different antibody out of it so that we can detect, and that antibody is basically attached to a bead. Okay, as shown in this particular figure. So this protein is being detected by this particular antibody that is attached to a bead. So what we'll do is we'll first see the DNA molecule. Okay, so this is the DNA molecule, and then what we do, we'll add this particular protein molecule in this, and then we immunoprecipitate DNA protein complexes using this beadwala complex. And then what we do after like separating out these particular complexes using this particular bead, then we separate out the protein and the DNA molecule. So this protein will be separated and the DNA will be separated. Then we identify the DNA using PCR. So various specific primers we'll use, and we'll identify the particular DNA molecule and sequence it. So now we'll get to know the sequence of the protein binding region on the DNA so that we can develop certain vectors like for cloning and other stuff. So this was the chromatin coimmunoprecipitation uh, assay through which we can detect the DNA binding region of the protein binding region on the DNA, and we can like identify through various means. Okay, so these were the four techniques, and then we'll move on to the next topic, that is regulation of transcription in prokaryotes. Okay, so now as we know that the transcription needs to be regulated. Okay, so there are as we have already discussed, there will be two kinds of genes. One is constitutive and the other one is inducible. So constitutive are the ones that are expressed at every time. Okay, and inducible are the ones that are expressed only on a specific that are expressed only once the signal is received by the DNA. Okay, so for the constitutive ones, we do not need any kinds of regulation because is continuously expressed. But for the inducible ones, we need to regulate their production and expression mechanism. So now there are certain mechanisms we'll study in how they regulate the process of transcription in prokaryotes. So first is the recruitment of the RNA polymerase. So as we know, there is a region called the promoter where the RNA polymerase bind. Okay, so like we are seeing here, this will be the -35 region and this will be the -10 region of the promoter. Okay, so here the RNA polymerase basically binds, and there are two more sequences that are activator binding sites and the operator. So as we already discussed about the different kinds of promoter, one is strong and one is weak. So if the promoter is weak, it requires other elements like activators so that it can efficiently do the transcription. Okay. So activator binding site is needed for basically the weak promoters. Okay. So activator binding site is this one that will be just before the promoter, and then we have the operator. So operator is the region where the repressor binds. Okay, so as we can see in the diagram B. So if the repressor can bind to this particular operator, then what will happen? The RNA polymerase cannot bind here. So this means this leads to no transcription. But in this case, if the RNA polymerase is binding to the promoter, this is the ideal scenario where we'll see basal level of transcription. Okay, now what will happen, the repressor if the repressor is bound, then there will be no transcription. But if we have the weak promoter and we need another protein for the RNA polymerase to bind efficiently and continue the process of transcription, then we have the activator molecules. So these activator basically increase the activity of the RNA polymerase. So they help in binding as well as the efficient or activated levels of the transcription. Okay, so this was first mechanism where the transcription is regulated, that is at the recruitment of the RNA polymerase. Then moving on to the allosteric binding of activators. So like as we already saw that activator binding site is needed basically by the uh, weak promoters. So now when RNA polymerase binds to a weak promoter, then sometimes what happens, due to some inhibition or any other signal, the transcription cannot function. Okay, this example we'll see further in the lac operon also, that how it helps in the transcription. So what happens in this case, if an activator is being bound before the RNA polymerase, so it will help in binding to the promoter and further help in transcription. Okay, just now we discussed uh, the role of activators. So these basically activate or stimulate the activity of the RNA polymerase. So now moving on to the third type, the DNA looping. So what happens? There can be activator side or repressor sites that are too far from the RNA polymerase. So for example, we have this long DNA. This was the promoter where the RNA polymerase is about to bind, and here is the activator or repressor site. So what will happen if the activator has to bind to the RNA polymerase, then this particular area has to be looped out or it has to be bent so that this activator can directly interact with the RNA polymerase. So this is the scenario where the activator site is basically near to the uh, promoter site. So there no looping is required. But if the activator site is far away from the promoter, then the looping is like required so that this particular activator molecule can interact with the RNA polymerase. And here the DNA binding protein is required as shown in this figure. DNA binding protein is required so that activator basically interacts with the RNA polymerase. This feature is basically a feature of the eukaryotes. Okay, where most of the promoters are weak and we need various activator molecules or enhancer molecules to increase the activity of the RNA polymerase. But in some prokaryotes that are more related to the eukaryotes, this particular scenario is found. Okay, so this was the third technique, that how the transcription is regulated in the prokaryotes. The first was the binding of the repressor, the second was the activator molecule, and third one was the looping of the DNA. Okay, so now we'll move on to the operon model that is present in the prokaryotes. Okay, so starting with the operon model, so this is only found in the prokaryotes. Okay, so what was the basic need of the operon model in prokaryotes? So these are small organisms. Okay, so if like different genes are expressed during different time, so that will increase their energy requirements. So to minimize the energy requirements, okay, so to minimize the energy requirement, the nature has devised a mechanism so that different genes that have similar function or they are involved in a particular process are linked together and they are connected to a same promoter. Okay, so that means it will form a single transcript that will have the transcripts of different genes that are transcribed. Hence the RNA is polycistronic. Different genes transcript are linked together because they have a common promoter, and then during translation they produce different proteins. Okay, and then what will happen if they are involved in a single process, so that they can coordinate in the regulation also. Okay, so this was the operon model that was given by Jacob and Monod. Okay, and they basically found out that the process involving different proteins uh, are being like present together in a DNA, so that only a single RNA can be formed and leading to minimal energy requirements. Okay, so the different genes that function together are placed together so that they are produced when only the signal is that they are needed in a process. That's the simplest explanation. So what we have is we have a regulatory gene so that it will control the expression of all these particular genes, and we have a common promoter which will produce only the single mRNA that will be yielded of these particular genes, and during translation it will have separate ribosome binding site so that they can produce different proteins out of this single mRNA. Okay, so this was the basic idea about the operon model. And now what we'll do is we'll be taking some examples about the operons that are being well studied. First is the lac operon and the other one is the tryptophan operon. So we'll be starting with the lac operon or the lactose operon. So this particular operon involves genes involved in metabolism of lactose. So this is widely studied operon model that is like a hallmark in the molecular biology. Okay, so first we'll discuss about the different genes that are present and involved in the regulation. Then we'll see the different mechanisms of regulation present. Okay, So first we'll see about the repressor gene. So this particular uh, gene sequence that I have marked is for the repressor. This is the regulatory gene. Okay, so it has a separate promoter which leads to the production of the mRNA and further the the protein. So LAC-I is the repressor, okay, or inhibitor of this particular operon, and this is the operator. So we have three operators here. First is the main operator, where this LAC repressor that is produced binds. Okay, so this particular operator is the main region where the repressor binds, and then comes the two operators O3 and O2. So O3 is also involved in the regulation of like binding of the repressor. Okay, and how it will bind, so we'll see a bit later. And regarding O2, the information is a bit less, but it has been seen if we mutate this particular O2, then the transcription also halts. So it is under study that how O2 is involved in the regulation of this particular um, operon. Okay, then this is the case with the operators. So operators are basically the repressor binding sites, as we have seen in the previous slides. Then comes the promoter of this particular gene sequences. Okay, so this particular promoter is the binding site of RNA polymerase, and it's a weak promoter. Okay, so how it's a weak promoter, we'll see in a couple of slides. Now, when there are three main functional genes that codes for proteins. So one is the LAGZ that codes for an enzyme called as beta galactosidase. Then we have a LAC-Y that codes for lactose permease. And then we have LAC-A that codes for galactoside transacetylase. Okay, so talking about the LAC repressor protein, so it has a helix turn helix motif, so that it will directly bound to the operator, and it generally binds in a homotetramer form. Okay, so these were the information about the sequences that are involved in the LAC operon. So we'll go a bit in detail so about the structural gene or the functional genes that are present here. So first is the beta galactosidase. So what it does is it basically converts lactose. Okay, so it's a disaccharide. So it converts lactose to galactose and glucose. Okay, so these particular both the sugars will enter the carbohydrate metabolism. Okay, and uh, then we have the lactose permease. So this particular enzymes basically helps in entry of lactose inside cell. So this basically is a structure of the lactose permease, and it how it does is it has a secondary, it is secondary active transport because it also like uh, needs a hydrogen ion to transport the lactose inside the cytoplasm. So what happens, it will translocate cytoplasm through through the protein only when one hydrogen ion is attached to the protein. So this is a classic example of the active transport, where for transferring one molecule of lactose inside the cell, one molecule of hydrogen ion is also needed. Okay, so this is about the lactose permease. It allows the entry of lactose inside the cell. Then we have galactoside transacetylase. So what it does is it basically transfer the acetyl group from coenzyme A, the donor of acetyl group, to a galactoside molecule. Okay, so this particular enzyme is not involved in the metabolism of the galactosides, but it is mainly involved in the detoxification process, not only of the galactoside but also of the different drugs and various other organic molecules. Okay, so this was the idea about different enzymes that are being produced by the lacoperon that are involved in the degradation of the lactose so that the cell can utilize it. Now we'll move on to the regulation, how the regulation of lacoperon takes place. So as I've already told you that uh, as I've already told you that whether when this lacoperon is switched on only when lactose will be present. Okay, so what will happen if lactose is present? Then only these enzymes will be formed and these structural genes will be activated. And if the lactose is absent, then in that case, this there will be repression in the like the genes will not be expressed and the proteins will not be produced. Okay, so what will happen? This lac-I, as I told you, will produce a repressor monomer. Okay, and then it will in a tetramer, it is activated form and it will bind to the operator region that is near to the promoter of the structural genes. Okay, so now what will happen? This will create hindrance in the way of the promoter. Oh, sorry, RNA polymerase. So RNA polymerase cannot move through this operator so that it will not be able to do the transcription. So this was the scenario when lactose is not present, so the lacoperon is repressed. Okay, so hence the lacoperon is repressed. Now what happens when lactose is present? So it enters the cell, and what will it do is it will directly bind to these uh, repressor molecules and it will bring about a conformational change. Okay, so what will happen? It will be released from the operator, and the RNA polymerase can directly perform the transcription of these structural genes. Okay, now these structural genes will be translated to different proteins that are involved in lactose metabolism. Okay, so when lactose present, then there will be no repression of uh, the lacoperon and different genes will be produced, and when lactose is not present, then there will be no genes produced. Okay, because RNA polymerase cannot move through the operator. So this was the scenario that was studied earlier. So this was the first simple mechanism of regulation, that how it happens. Now moving on. So what is the basic inducer of the lacoperon? Okay, so it is actually allolactose. This is the inducer of the lacoperon. So, if anyone asks you the question that whether lactose is the inducer or allolactose, so allolactose is the basic inducer of the lacoperon. Okay, not the lactose. Lactose is comes on the second number. So first is the allolactose. it is the isomer of lactose only, and sometimes the lactose is converted to allolactose using beta galactosidase only. So what happens, the for the cell to convert this lactose to allolactose, first it has to bring about the lactose inside the cell. So what happens, there is a concept of leaky expression that I talked about, that it is a weak promoter. So what happens during normal scenario, there is some basal level of expression of genes happens inside the cell. So what this does is this will produce small amount of all these lag Z, lag Y and lag A genes. Okay. So although this is highly regulated, but this small amount is present that leads to leaky expression. Okay, so this was the phenomena that was like hypothesized, and it has been proved till now that this leaky expression only leads to the production of some amount of beta galactosidase, so that the lactose that is present there can be converted to allolactose, and that is the main inducer of the lac operon. Okay, now the same thing applies with the permease, that is lactose permease. So small amount of lactose permease, sorry, so small amount of lactose permease is present on the cell membrane so that it will allow the lactose to be inside the cell. Okay, so that is why the operon can be induced in this way. Now moving on to the utilization of lactose, that can be a choice or like that leads to catabolite repression. So what happens during normal scenario, like this is the cell and it is present in a medium that will have more than one sugar to take up. Okay, so one would be simpler than the other. So what will happen, the cell would try to take the simpler sugar first, then it will take the complex sugar. Okay, so this is the thing that is observed in E. coli when it was grown in glucose as well as lactose. Okay, so here two like phases of growth were observed, and this was seen that where first the glucose was used as a carbohydrate source, and then lactose was used as a carbohydrate source. Okay, so this like two plateau curve is called as dioxic growth. Okay, so this was seen in various cases where the simple sugar is being utilized by the bacteria first as compared to the complex sugar, and this is seen in various other cases also, like arabinose and other sugars also. Then what happens when glucose is being utilized, then all the genes that are involved in the lacoperon are switched off, so hence the lacoperon is switched off, and when lactose is used, then the lacoperon is switched on again. So there is another mechanism that basically helps in the regulation of the lacoperon in the presence of glucose and in the absence of lactose. Okay, So what happens when glucose and lactose both are present? The lac operon is switched off, and only a basal level of transcription is being observed in this case. Okay, and then what happens if glucose is present and lactose is absent? Then there will be no transcription. But if if only lactose is present, then the activated level of transcription was observed. And in this case also, it is basically repressed. Okay, that is too less for the cell to utilize the lactose first. Okay, so this scenario is basically termed as catabolite repression. Okay, so because the utilization of first one catabolite, okay, basically represses the utilization of second catabolite. So hence catabolite repression, the term was given. Okay, now moving on. So now moving on to the regulation of the lac operon, the complex version. So what happens? This, as I told you, that lac promoter is a weak promoter. So it requires a activator also to activate the process of transcription for this particular lac operon. Okay. So what happens? There is a site called as cap site, that is the binding site of a protein called cap or CRP. So cap is basically catabolite activator protein, and CRP is CMP receptor protein. So what happens? This particular protein binds to the cap site and it's activated in the presence of CMP. So when CMP binds to this protein, it will bind to the capside and it will as act as an activator of this particular RNA polymerase. So this RNA polymerase is the prokaryotic RNA polymerase that we have discussed in the last class also. Okay, so this basically increases the binding of RNA polymerase to promoter. This was the first thing in the complex reaction. Then comes the role of operators. Okay, so these were the three operators that I was talking about earlier in the lac operon, that these three operators are present. Out of these O1, this is the sequence of these three operators, and it has been seen that like if either of the operator is mutated, then the level of repression on the lac due to lac I was observed, and if we have all the three operators in line, then it will yield a maximum repression by the lac I. If we have only the O3 and O1 repress operators, that will decrease uh, the repression to this much amount. So this is basically one third of this particular thing. So that means O2 is also important for regulation of the lac operon. Then if we do not have O3, then it was literally half, nearly half of the when three operators are present. Okay, and when only O1 is present, then it was like 18. So this was minimum. Hence, it was shown that if all the three operators are there, then this would show maximum repression by the lac I. Okay, hence, what we'll see in the next slide, that how O3 and O1 are linked to each other during this particular uh, com- uh, complex regulation of the lac operon. So now what happens when the lactose is not there, lactose is absent and glucose concentration is high. So the first mechanism of the lac operon, that is the regular mechanism, functions as it is. So what happens? This lac repressor will bind to the O3 and O1, that is operator 1 and operator 3, and it will form a loop kind of structure. Okay, so what is the importance of this particular loop is that the CRP binding site that is just adjacent to the operator 3 and near to the promoter, that can come close to the promoter, more close to the promoter. Okay, so that it will help binding the RNA polymerase. So this was the basic need of the R, and other studies also suggest that if this loop is formed, this particularly creates the CRP binding site. Okay, so when CRP is linked to CMP, it recognizes this particular site and binds, and then it releases this particular loop, so that now it can bind to the RNA polymerase. Okay, now here we'll see two phenomena that if and one more thing before I move on. So, the regular repressor phenomena, that when lactose is present, then it will act to the repressor, it will bind to the repressor, and it will allow the operon to be functioning. So, this mechanism also acts simultaneously with this mechanism. Okay, so this is actually the more quality control kind of mechanism that takes place inside the cell. So now what happens? There can be two scenarios. One where the glucose is present and lactose is absent. Okay, so where here we have four scenarios basically. Sorry, three scenarios where glucose is present and lactose is present, where glucose is present but lactose is absent, and where glucose is absent but lactose is present. Okay, so and one more scenario where glucose is also there, not there, and lactose is also not there. Okay, so we'll see all these scenarios and how the CMP and the operon functions. Okay, so first we'll see the scenario where glucose concentration is high and the lactose is absent. So when glucose concentration is high, so it will form more ATP. ATP number will be high. Okay, but what will happen next is it will decrease the amount of CAMP, that is cyclic AMP, because the metabolism is high. So the amount of energy will be converted to form ATP rather than CAMP. So when glucose is high, then the amount of CAMP will be low. Okay, so this is the case, and what will happen now? The and lactose is absent. So here the lac repressor binds to the operators, developing a loop so that this CRP binding site has been created. Okay, and here the lac operon is off. Okay, so this was the first scenario where glucose concentration was high and lactose was absent. Okay, in the second scenario, what we see is where glucose concentration is low and lactose of concentration is also low. Okay, lactose is not present. So what will happen? The CMP levels is high, but due to absence of the lactose, there will be no expression of this, or no, the lacoperon will be off in nature. Okay, but as CMP is high, because this metabolism is low, so ATP concentration will go down. This will lead to increase in CMP concentration inside the cell. So what will happen? It will bind to the CRP protein and go and bind to the CRP binding site. Okay, but this loop cannot be relieved because the lac repressor is bound as it is because of the like lactose is not there. So this was the second scenario. In the third scenario, when glucose concentration was high and lactose was also present. So in this case, what will happen? This When lactose concentration is, because both the metabolite are there, so metabolism will be high. So this means ATP amount will be high. So CMP will be low. Okay. So now what happens? As first we'll see the lactose one. So because lactose is also present, that means it will bind to the repressor. It will convert into allolactose and bind to the repressor, and repressor will be removed from the operator region. Hence, the operon will be on in nature, but it will show low level of gene expression because the second mechanism is off because of low levels of CMP. So here CRP cannot bind to the CRP binding site because the CMP levels are low. Hence, it will lead to only basal level of gene expression. So this is the case where in the last slide that we have seen when glucose and lactose both are present. So this is the case of the first plateau. Okay. So in this case, the operon will be on, but it will show basal level of gene expression. Okay. Now, this was the third scenario where both of them were present. Now, we have a scenario where glucose concentration is low and lactose is also present. So, what will happen? Although metabolism will be there, but it will not be that high. So, ATP levels will be marginal. That will lead to increase in levels of the CAMP. Okay. Now, as lactose will be present, so what will happen? This lac repressor will be bound by the allolactose. Okay, and it will cannot bind to the operator regions. Okay, it cannot bind to the operator regions, thus relieving the hindrance caused in the path of the RNA polymerase. Now, as CMP levels are also high, it will bind to the CRP proteins, thus, it will bind to the CRP binding site, thus, it will activate the RNA polymerase. Okay, so it is an activator. Now, the protein is also bound to the its site, so now it will activate the RNA polymerase so that it can transcribe this particular operon. So the operon will be on and it will show high level of gene expression due to binding of this activator protein. Okay, so these were the scenarios that were seen during the dioxic growth when glucose and lactose both are present. And this second mechanism is also important to yield large or the more amount of gene expression when glucose is absent and lactose is present, so that lactose is highly like quickly metabolized inside the cell. Okay, so this mechanism is more important as compared to the normal repression mechanism, and this also holds important point in the competitive examinations also. So this many questions have been asked for like involving this particular mechanism. We'll see those in the next few classes, that how the questions have been asked. Okay, now moving on to a phenomena, that is the lacoperon mutations or the mirodiploids. So, why we are studying these? Because of two reasons. First, these mutations were involved in the discovery of the lacoperon, and second is the case where sometimes what happens, the lacoperon is also integrated in the F plasmid. So, what will happen? It will lead to the presence of diploid because, or mirodiploids rather, because only for this particular genes, if the plasmid is present in the cell and the genome also has this particular gene, so only for this particular gene it will be deployed in nature. So it these are called as monodiploids. Okay, so now we'll study different effects of mutation on these particular genes, where it one part is present on the genomic DNA, one allele, and the other one is present on the um, plasmid DNA. Okay, and here we'll be using some terms. So we'll explain, then we'll move on to the mechanism. So first we see the recessive term, that means there is no change from the wild type as like the normal mechanism. Then we have the dominant. So dominant means that the phenotypic change that we are observing is different from the wild type. And cis means that mutation affects the same allele only. That means if this particular allele is producing a protein, that will act upon this particular allele only and not other alleles. And trans-acting means the protein that is being produced by this particular allele can act on different alleles also. Okay, so just a piece of information that the lac I codes for the repressor. Okay, so this is trans-acting in nature. That means it can also directly attach to different allele also. So now we'll see different conditions that how the repressor gene is mutated and what can it yield to. Okay, so now first thing is when the repressor gene is mutated, okay, in one of the alleles. So one have the positive repressor and the other have the negative or the mutant repressor. So what happens now, the gene that have the normal repressor will produce it, and it will act upon both the operators of the plasmid DNA as well as the genomic DNA. Okay, hence no lac products will be produced in the absence of lactose. Okay, so this will yield into the normal phenotype of the lac operon. So this leads to the mutation is recessive in nature, so that it will not be able to express itself. So this was the first scenario. In the second scenario, when there is a mutant operator, so there is mutation in the operator, so where repressor binds. So what will happen in this case, where only one allele has the mutant operator, such that the repressor cannot bind to the product. So what will happen? The repressor is fine; it will be produced as it is. Only one of the genes will have the operator sorry will have the functional operator where the repressor can bind, but the other have a mutant operator where the repressor cannot bind. So in this case, this particular allele will not produce any products in the absence of the lactose, but this particular allele will produce the lac products in the absence of the lactose also. So this function is not the wild type phenotype, hence it will be called as the dominant. And why cis dominant? Because the repressor produced by this particular allele cannot function upon itself, so this is cis dominant. Sorry, it will function upon itself, so this is cis dominant. Okay, this was the second scenario. Now moving on to the third scenario, where there is a mutant repressor gene. Okay, so mutant repressor means that in um, so then the other case, in the when there is mutation in the lac repressor gene, so in this case what happens, the sorry the repressor cannot bind to the inducer. Okay, so what will happen, even if the inducer will be present, it will not be able to bind to the repressor, hence the lacoperon is in switched off condition only. Okay, so this changes the binding pattern to the operator such that it is always bind to the operator rather than it is bind to the inducer. So what will happen, even if the inducer is present or absent, it will be switched off only. Okay, so in this case, the lacoperons will be uninducible, and this is also a dominant because it is away from the wild type. So it is also cis and trans dominant both. Okay. Now, in the other type of mutations where there is uh, like it cannot bind to the operator. So the repressor mutation where it cannot bind to the operator. So the conformational change is being made such that it is not able to bind to the operator. Hence, even if the lactose is absent, the operon will be on. Okay, so this leads to when in the absence of lactose, the operon will be in the on states, and we can observe different lac products. So here it is also a dominant negative mutation. Okay, so these were the four ways where the mirodiploids have been used for the uh, like discovery of the lac operon. Okay, now coming to the last part, so we'll see some applications and how it has been exploited for various applications. Okay, so first is ONPG, that is orthonitrophenyl beta galactoside. So this assay is basically colorimetric and it helps in the detection of the beta galactosidase activity. So what will happen if beta galactosidase is present? So what will it will do is ONPG. So it will degrade ONPG into galactose and orthonitrophenol, and this is basically yellow in color. So this can be detected at a particular wavelength in spectrophotometry or colorimetry. Okay, so this particular assay is for the detection of the beta galactosidase in the cell. Okay, and this assay is also basically like used for detection in a 96 well plate. Okay, so this is an amplified reaction of this particular activity. Then we have another component, 5-bromo 4-chloro 3 indolyl beta D galactopyranoside. This is in short called as XGAL. So what happens is when during cloning, what we do is we attach the transgene with the LACZ gene. Okay, so if or the LACZ gene, sorry, we have a marker like antibiotic resistance marker or the LACZ gene. So if we are cloning and the plasmid has this, so any bacteria that is taking up this particular plasmid, so the LACZ gene is if present and we add XGAL, so this produces two compounds after degradation. One is galactose and the other one is this 5 bromo 3 chloro 4 chloro 3 hydroxy indole. So this particular compound is blue color in nature. Okay, sorry, it is oxidized into 5,5-dibromo 4,4-dichloro indigo. So this is an insoluble blue colored compound. So this particular after adding xgal to the medium, the colonies that have the LAGZ gene will be in blue in color because of the presence of this particular compound, this indigo compound. So this is a basic mechanism of how we can distinguish the transformants with the non-transformants. So the transformants will have the plasmid with the lag Z gene, so they will show blue colonies, and the non-transformants will do not have the plasmid, so they will show white colonies. Okay, so this has been shown in the next slide also. So this is what I was talking about. So this is a marker gene. Okay, so if the sequence have been cloned in this particular region, then it will show like the laxate gene will be disrupted and it will not show any blue color after adding XCAR. So this will be the white colony. And if the gene is not inserted in this particular laxate gene, then that means the laxate is full intact, and this will convert the product into blue color. So X-gal can be converted and these blue colonies will be observed on the culture plate. Okay, and these white colonies that we see, that means the gene has been inserted in the LAGZ gene that is disrupted. So this is a insertional marker. Okay, insertional marker, and this is how it has been used like in cloning. And then we have IPTG, that is isopropyl beta D1 thiogalactopyranoside. So what it does is, so this is basically a molecular mimic of allolactose, that is the inducer of the lacoperon. Okay, so in various vectors it has been used as the lac I, and operator is used to induce the expression of transgene. Okay, especially in pet vectors. So here the lac-I is being inserted so that it will directly bind to the operator. The lac-I and operator system has been exploited in this case, and in such a way that the transgene cannot be expressed before the adding of IPTG. Once we add IPTG, then the transgene is expressed. Okay, so this is another example where this particular Lacoperon system has been used. This is basically the basis of the regulation. Okay, so this is in the recombinant expression that we have discussed just now about the blue-white selection method. So this selection method is called as blue-white selection method for selecting the recombinant proteins. Okay, so this comes to the end of our lecture, where we have discussed the lacoperon, its regulation using simple means with the help of repressor, and the complex means with the help of the activator proteins, and how these mechanisms has been employed in usage of various other cloning reactions. Okay, now, if you have any doubts, you can either write in the comment box or you can ask me in the next lectures. Okay, so thank you.